Great. Well, thank you to the organizers for inviting me to this fantastic conference. I think my talk will follow nicely from David's. Originally, a few years back now, the plan was to talk about derivation and use of epicardial lineages from human pluripotent stem cells. I've tweaked that title just a little bit to talk about pro-regenerative properties of human stem cell derived and human fetal epicardium. So I can show you some of our latest data on human fetal epicardium. So this is a developing chicken heart, and you can all see the epicardial layer really nicely. This is a mesothelial layer on the surface of the heart. And we know that in development, this mesothelium undergoes EMT, and the cells migrate into the subepicardial region. They're termed epicardial-derived cells, or EPDCs. These give rise to fibroblasts, coronary smooth muscle cells, and there's tremendous crosstalk in development between the epicardium and the underlying cardiomyocytes and developing blood vessels. We know that in the healthy adult heart, the situation is much more quiescent. The epicardium is quiescent, but after injury, we see reactivation of the epicardium, which to some extent resembles what we see during embryonic development, but it's not quite the same. We see some limited EPDC migration, there's some angiogenesis, a lot of matrix accumulation, but limited or no cardiomyocyte proliferation. We also know that in species such as zebrafish, which regenerate their hearts as adults, the epicardium is required for this. And this is data from Ken Poss's lab, where when he ablated the epicardium, the adult zebrafish lost the ability to regenerate the cardiomyocytes. And then when the epicardium was allowed to recover, then the cardiomyocyte regeneration then restarted. So in our lab, we've spent some time generating a range of different cell types from human pluripotent stem cells. And we published a protocol some years back to generate epicardium from stem cells. And this basically consists of early mesoderm induction, followed by patterning to lateral plate mesoderm, and then treatment with Wnt, retinoic acid, and BMP4 to give us an epicardial state. And this epicardium in vitro has this lovely cobblestone appearance. It looks very much like human fetal epicardium if you were to culture that. Expresses a whole range of epicardial markers. And given the right stimuli, we can make these epicardial cells form either coronary smooth muscle cells or fibroblasts. So given what we know from development and the crosstalk between epicardium and other cell types, we wanted to see if our epicardial cells would recapitulate that. So we made engineered heart tissues using human pluripotent stem cell-derived cardiomyocytes or cardiomyocytes plus the epicardial population. And you can see that when we made, this is using a collagen gel, just cardiomyocytes here, we get contraction. This is calcium flux you're seeing. It's not particularly good, it's somewhat heterogeneous. When we add epicardial cells to that engineered heart tissue, we get much better contraction, much more homogeneous electrical activation. And if we do some measurements, we can show that we get increased force of contraction with the epicardial cells in there, and we get much better electrical activity as well. And we compared it here to adding in either primary human mesenchymal stromal cells or stem cell-derived stromal cells. So our epicardial cells in our hands seem to do better than other cell types. Well, this was very encouraging in vitro. The question was, would epicardial cells have a similarly beneficial role in vivo? So we looked to see if we could remuscularize the heart using both epicardial cells and cardiomyocytes. And this work was done in collaboration with Chuck Murray. And this is using the athymic rat MI model. So the rats undergo ischemia reperfusion injury. 4 days after that, they have cell injection. And the groups were either epicardial cells alone, cardiomyocytes alone, the combination of the two cell types, or no cells, just vehicle only. And we studied these rats at 4 weeks and 3 months after injury. And what we saw at 4 weeks was that the cardiac grafts were much bigger in the group that had epicardial cells as well as cardiomyocytes. In fact, they were 2.5 times bigger than when we just put in cardiomyocytes alone. In addition to that, we saw increased vascularization of the grafts. Here you can see CD31 staining of our grafts, and there was increased vascular density when we had epicardial cells present in the grafted cells. We also saw improvements in cardiac function. The dotted line, basically, is the function at the point of cell injection. And if you don't put any cells in with the gray bar, you can see that the ventricular function continues to deteriorate over 4 weeks in these rats. Epicardial cells alone, in green, didn't make any improvement to that. But when we put in cardiomyocytes, we arrested that negative remodeling. And that's data that Chuck Murray and Mike LaFlamme have previously shown, that the cardiomyocytes prevent that negative remodeling. What we showed was that the combination of epicardial cells and cardiomyocytes actually improved cardiac function. And we think this is due to the increased size of the grafts, the increased vascularization, and the fact that the cardiomyocytes within the grafts appear to be more mature. The cells we put in persisted for 3 months. This is a human mitochondrial antibody, and you can see whether we put it in whichever group we looked at, whether epicardial cells alone, the cardiomyocytes, or the cell combination group, the human cells persisted in the rat up to 3 months. And the functional benefits that I've shown you at 1 month persisted at 3 months as well. So fairly long-term benefits, at least in the rat model. Now, what's the mechanism of this interaction between the stem cell-derived epicardium and the cardiomyocytes? And that's something we're very interested in as a lab. I'm not going to tell you any more about this now because we have a short talk coming up looking at the particular role of the matrix and fibronectin in mediating this interaction. Now, my students, Lei Ping Ong and Jonathan Lee, have been working on some of this mechanistic aspects. Unfortunately, they couldn't be here, but one of my postdocs, Vince Knight-Shriver, who's in the audience, will present that work later on. So I'm not going to talk any more about that now. This is Vince. He's also a computational biologist, so he's done a lot of work looking at fetal and adult epicardial cells. I'm going to switch gears now to tell you a little bit about our work on that. We've taken human fetal hearts at these time points, between 8 to 11 weeks, and we've looked at an apical section and also part of the base and done single-cell analysis. We've then combined that data with data taken from the Human Cell Atlas. This is Sarah Teichmann's work looking at normal human hearts, and we've taken both single-cell and single-nuclear data from 6 normal human hearts. If we combine all that together, what we see is, much as David said, that the cell types we have the same cell types in both fetal and adult hearts. Although interestingly, we do see this large orange population here, which is difficult to classify, and we've just called it a sort of immature cell type. But most of the other cell types are shared fairly well between the fetal and the adult hearts. We see the epicardial population here, and subclustering showed us 3 different populations of epicardial cells. So 8, 9, and 10. So if you look closer at these, we named these mesothelial epicardium, fibroblast-like epicardium, and proliferating epicardium, and I'll show you why. So all of the epicardial cells that we see share this range of epicardial markers, but the fibroblast-like epicardial cells also express fibroblast markers and also show signs of EMT. So what we think we're seeing is 2 different populations of cells. One is a mesothelial population, and also this fibroblast-like population, which we think represent the EPDCs that have undergone EMT. Now, the distribution of these cells is really interesting. So if we look at the distribution of cells in the fetal or the adult hearts, most cell types are shared between the fetal and adult hearts. If you zoom into the epicardial cells, you can see that the mesothelial epicardial cells are found equally in both adult and fetal hearts. But the fibroblast-like epicardium is found almost exclusively in fetal hearts. The same actually goes for the proliferating epicardium, but there's very few of those, so I'm going to focus mainly on these 2 cell types. So we think that we've got these 2 cell populations, and the difference between the adult and the fetal hearts is that the fetal hearts have a lot more of these epicardial-derived EMT type cells. Now, this is all bioinformatically. Do we have any spatial evidence that this is actually what happens in these hearts? So this is some spatial transcriptomic data from Sarah Teichmann's group that we're collaborating with. And what we have to do, if we look at genes expressed in the mesothelial population, we see them just on the periphery, on the sort of where you expect the mesothelial epicardium to be. If we look at the fibroblast-like genes, we see them on the periphery, but also in the regions just below that, corresponding to where we might expect to find EPDCs and cells migrating into the epicardium. So I think this model is correct, and this is what we're seeing with these cells in both adult and fetus, and this cell population just in the fetal hearts. We subclustered these. I'm not going to go into too much detail, but only to point out that if we look at a primary component analysis, primary component 2 categorizes these by age, so we have adult cells here on the left and fetal cells here on the right. And if we look at gene modules, in other words, genes that are co-expressed, we found about 6 different gene modules, and these gene modules depended on the cell type and the age of the sample. So module A is expressed mainly in adult cells, and we can see that this, if we cluster our samples by age, these genes are expressed at low levels in the fetal epicardium and then increase with age. Whereas modules C, D, and E are genes, modules that are expressed primarily in the fetal hearts, and these are upregulated in the younger samples and then decline with age. So what do these modules tell us about what's going on in terms of the processes? So what we see is that, for example, here in modules D and E, these are regenerative programs. So we see things such as blood vessel development, tissue development, animal organ morphogenesis, differentiation, and so on. And these regenerative programs are lost with age, and they're not expressed to a significant extent in the adult epicardium. We did a lot of study. I'm not going to show you all the details, but basically, what are the actual pathways involved in these regenerative programs? So cell phone DB analysis showed that the epicardial cells communicate with a range of cell types in the developing heart, but very predominantly with the underlying vasculature. And the mechanisms for interaction that really came up were Wnt2b, NRP2, TGF-beta3, and a range of different collagens. What about the adult program, this module A? What really surprised us was module A had a lot of Go terms that were associated with stress, defense, and immune response. So if you look at some of these, we've got response to chemicals, response to bacteria, response to stress. And it seems that the adult epicardium, at least compared to the fetal, is primed for stress and immune activation. And this was really quite interesting and surprising to us. Okay, how does our samples compare with, the fetal samples compared to what we're generating in vitro from our stem cells? So we looked at our stem cell model at a range of different time points, and using single cell analysis, we can see we have 2 populations, one that expresses PODX cell and BNC1, which we've described before, and TI1 and TCF21 here. And this PODX cell BNC1 population is, if we use an unbiased prediction algorithm, looks very similar to the mesothelial epicardium, and this other population here is very much fibroblast-like. So we think that the stem cell-derived epicardium is very similar to the fetal epicardium that we're seeing in human fetal hearts. The gene modules that I showed you, here this is expression of those of either human adult epicardium in blue, the fetal epicardium in yellow, or our stem cell-derived epicardium in green. If we look at module A, which is expressed in adult, you can see that our stem cell-derived epicardium doesn't express that. Modules C, D, and E, which are expressed primarily in the fetal epicardium, not the adult epicardium, are also expressed by our stem cell-derived epicardium. So we think our stem cell epicardium models human fetal epicardium reasonably well. I'm going to finish there just to conclude and summarize. What I've shown you is that the stem cell-derived epicardium promotes cardiomyocyte function-engineered heart tissues and has a similarly beneficial effect in vivo for remuscularization and neovascularization in a rat-MI model. I've shown that the native human epicardium loses the regenerative modules, but does acquire genes that are related to stress and inflammation as we go from the fetal state to the adults. The stem cell-derived epicardium shares features with fetal epicardial tissues, and what we have hopefully here is a roadmap that might be used to restore regenerative human epicardial function. And really my final takeaway message is what I started off with, which is that the developmentally early epicardial state in humans is highly regenerative, and this might have implications for cell therapy in terms of thinking about combined cell therapy or endogenous regeneration by perhaps reactivating the fetal state. I just want to thank the people who've done the work, the people in bold, or the people who have done most of this work, and the people who have funded it. Thank you.